Okay, so here we are on the second piece of the reproductive system lecture, and now we're going to go into the female reproductive system lecture. So we already did the male, so I want you comparing the two as you go through, look at what's the same and what is different. And we're going to look, I'm going to start with a little bit of anatomy review for those of you who kind of forgot all that stuff. So the gonads for females would be the ovaries, and the ovaries are where the oocytes or the eggs are going to be produced, and they're also the location where you're going to produce the two important sex hormones, which are going to be estrogen and progesterone. So the ovaries, again, are a mixed gland, both endocrine and exocrine products. The exocrine product would be the eggs or the oocytes, and the endocrine product would be estrogen and the progesterone. One thing that I'd like you to notice that is different right off the bat is that for females, for males, remember, everything was kind of held constant through adult life, so constant level of sperm production, constant level of testosterone levels. For females, those are going to be cyclical, so they're going to increase and decrease as we go through the female cycle. So the ovaries will produce the eggs, and the secondary oocyte is going to be then kind of spat out from the ovary, and it'll be collected by the uterine tubes, okay, also known as fallopian tubes, if you know that term as well. So it'll go out from the ovary, just sort of be spat out, and then the little fimbriae, the little like waving fingers at the end of the uterine tubes will collect it, suck it up with a little current, and then the oocyte is going to travel along the uterine tubes towards the uterus. Here is the uterus here. Once it's in the uterus, it's going to implant in the endometrium. So what we're looking at here is a cross-section through the uterus here. And the uterus wall has 3 layers. The innermost layer, that is the endometrium. And the endometrium, especially in certain parts of the female cycle, it has all these glands and blood vessels, so it's nice and wonderful and juicy, wonderful place for the little, hopefully, you know, from the little embryo, if it is fertilized, to embed itself in. Okay, so that's where it would embed itself into there if it becomes pregnant. Underneath that, we have the myometrium, and anytime you see the word, the prefix, like the beginning myo means muscle. So the myometrium is made up of smooth muscle, and the major function of the myometrium is to contract during labor, to force the fetus out. And then the other thing that it does is during menstruation, what it does is it does little spasms of the smooth muscle to help shake the endometrium off, because the endometrium will be shed during menstruation. Okay, so the myometrium does those little contractions to help shake it loose, and that's why you get cramps. You get cramps in the myometrium there, for those of us who get that. And then surrounding the outside is the perimetrium, and the perimetrium is just the serous membrane layer on the outside for protection. Okay, so just to reiterate, make sure you get this piece. The endometrium is where the embryo would embed, and it is also the layer that will be shed during menstruation. Okay, let's look at those ovaries. Inside the ovaries, remember, that is where you're going to be producing your eggs, which are called oocytes, and this is also where you're going to produce estrogen and progesterone. So the eggs are going to mature inside of circular structures called follicles. Follicles is just a generic term that they often use for circular structures. So here is a follicle, and these are also follicles. Okay, so here we have a bunch of supporting cells, which are called follicular cells, and they're the ones, the follicular cells are the ones that are going to produce estrogen. Okay, so we can see the progression of the follicles. Okay, so there's a primary, and then secondary, and then almost mature ones. Okay, so now let's talk about oogenesis. And as I go through oogenesis, which is the process of making those oocytes or the eggs to produce the female gametes, I want you thinking about, well, what's the same as what we've already seen, and how is it different from the male spermatogenesis? Okay, so just like before, we're going to produce stem cells, and the stem cells will reproduce, make a whole bunch more stem cells using mitosis. That's what that's about. But when we're looking at meiosis, here's the process of meiosis here. So what happens for females before birth is those oogonia, which are the stem cells, they start the beginning step, so prophase 1 of meiosis 1, so they begin to make more oocytes, but they're going to be arrested. And what I mean by arrested is it just stops. It's just frozen in time. Okay, so at birth, the females have an ovary that's just full of primary oocytes. Okay, so they've undergone prophase 1, but they're just stuck in prophase 1. And an important thing to understand is that for females, all of the oocytes that you're ever going to make are already present in the ovaries. Okay, so they're going to undergo prophase 1 and then just sit there waiting their turn. And then only one of those arrested oocytes will be selected to continue to mature. Okay, so one of them will finish the meiosis 1, and I'm going to talk about how just one is selected in a minute. So one will be selected each month. The rest of them will either die off for another reason, or they'll just stay arrested in prophase 1. So the one that will be chosen will finish off meiosis 1. It'll go into meiosis 2, but then again, it'll be stuck in meiosis 2. Okay, so it finishes off meiosis 1 and it's stuck in metaphase 2 of the second division stage. Okay, and so the egg that will be ovulated, which means that you eject the egg out of the ovary, the one that will be ovulated is going to be a secondary oocyte stuck or arrested in metaphase 2. Okay, so we have a second arrested phase, and it'll stay in metaphase 2 unless something specific happens. So it will only finish the rest of the phases of meiosis 2 if it is penetrated by a sperm. So when it is penetrated by a sperm, at that point, it'll undergo anaphase 2, telophase 2, and it'll finish meiosis 2. And at that point, then you'll have the zygote. Okay, so you don't actually create the full finished product unless the sperm penetrates it. So if you don't become pregnant, the one that would come off during menstruation, just sort of come out of your body, or sometimes they get degraded before they even come out, that would still be stuck as a secondary oocyte in metaphase 2. You don't get a finished, fully functioning, real gamete, which is called the ovum, unless the sperm penetrates it. And then from there, once they're fully finished, they'll combine together and create a zygote from there, and then an embryo, then a fetus, and so forth. Okay, so let's look at what's different. Number 1, 2 phases, 2 arrested phases. First, before birth, they're all going to be arrested in prophase 1, and they will continue to be in prophase 1 unless they are selected to be ovulated. Okay, and then as they develop, they'll go into the second phase, the second meiosis, and they'll be arrested again in metaphase 2. And then they don't even finish completing metaphase 2 unless they are penetrated by the sperm. And if they are not penetrated by the sperm, they'll either die off or come out during a menstruation. And if they are, they'll finish up meiosis 2, become an ovum, which then combines with the sperm and so forth. Okay, so 2 arrested phases. This big old long process, 2 arrested phases. The other big important difference between spermatogenesis and oogenesis is look what happens here during the divisions. Instead of getting two equal cells out of it, like we saw in spermatogenesis. Instead, one of the cells hoards all of the cytoplasm. So you get a big, beautiful secondary oocyte, nice and big and round and wonderful. And then you get a little dinky one. That little dinky one that's very small, kind of sickly, that is the polar body. And polar bodies are not viable. They cannot make any gametes from it. They will not grow up to be an offspring. Okay, so even though they contain all the genetic information, they do not have enough cytoplasm to be a viable gamete. Okay, so you only get one there. And then when the sperm penetrates and it finishes off the second phase of meiosis 2, it happens again. Okay, so here we have another division here. And again, one takes all of the cytoplasm for itself. So you get one beautiful, wonderful ovum that's got all this wonderful cytoplasm. It's really, you know, a lot of good cytoplasms. It'll last a long time. And again, you get an ugly little small polar body that's good for nothing. Okay, so how many viable gametes do you get out of one phase of meiosis, one process of meiosis? Only one. Okay, a lot fewer than 4, which is for spermatogenesis. So keep that in mind. Okay, so let's look at how this relates to ovulation. So here's an ovary right here. And you'll notice that when you look inside somebody's ovary, you're going to see a bunch of cells, a bunch of eggs in multiple different phases, right? So they'll all be kind of in multiple different phases. Most of them will be in the primary oocyte. So remember, if you look at this page, most of them, just a big old collection of primary oocytes that are, you know, really small. Those are going to be found in what's called, sorry, they're right here, a primary follicle. Okay, so it's a primary oocyte inside a primary follicle. They're arrested in prophase 1. They're just sort of stuck in time waiting their turn. And then it's a collection of them are going to be, you know, are going to get enough of the follicle stimulating hormones to begin to develop. Okay, and as they develop, the first thing that happens, even before they, you know, continue on, is you're going to create more of those follicular cells, the follicle cells that surround the outside, and that's going to create what's called a secondary follicle. Okay, so the secondary follicle, and then you'll create more and more and more of those follicular cells, and you'll get bigger and bigger and bigger. And what actually begins to happen is now you start developing this empty space. The empty space is called an antrum, and so these are called antrum follicles. Okay, please note that inside the cell finishes meiosis 1 and it goes into meiosis 2, but it's going to be arrested again in meiosis 2. Okay, so in meiosis 2, that's now called a secondary oocyte. Okay, so once you have the mature follicle here, notice it has that space in here. That's because the egg kind of moves to one side. And what happens during ovulation is that the follicle ruptures open. Okay, so this whole kind of antrum space ruptures open, and that's just going to spit out this secondary oocyte. Okay, so here's your secondary oocyte. It does have some follicular cells kind of protecting it on the outside there, but there, inside there, we have our secondary oocyte, which is again arrested in metaphase 2. When will it become a full functioning ovum? Not until the sperm penetrates it. Okay, so our secondary oocyte will be spit out, kind of ovulated, that's the word for it, just sort of stuck out into the world, into the abdominal cavity. And that is our secondary oocyte, which is arrested in metaphase 2. Okay, so now what I want to do is take a little bit of a moment and let's compare and contrast the female and the male reproductive strategies. Okay, so first I want to look at how long it takes to get that functioning gamete. Okay, so remember that males from the start of puberty through pretty much the rest of their life, they're going to be continuously producing those gametes. And so if you look at the time it takes from the stem cell all the way through the entire spermatogenesis process until you get a fully functioning sperm, it takes about 74 days. Okay, so not a whole ton of time. And remember, this is constant. But for females, because you have all of the primary oocytes that you're ever going to have, and they're all just sort of sitting around waiting their turn, only one of them will be selected to continue maturation and to reach the secondary oocyte stage. And only from there, some of them will be, fully penetrated and become full functioning gametes. That means that it could take like all the way up until menopause for them to be fully functional, right? So these huge spans of time. Okay, so that if you look at this one right here, so very quickly, constantly, that means you can make quite a lot of sperm, okay? Over 1 trillion different sperm within one lifetime, whereas the females, it's going to be less than 500. Okay, so you don't have nearly as many, like a tiny fraction of the number of total gametes produced in a lifetime, and that's for 2 reasons: the time span that it takes, and also the fact that for each cycle of spermatogenesis, you get 4 wonderful equal sperm, and for the females, you only get 1 single ovum, and then the other ones are just going to be non-viable. Okay, so it takes a long time, and you don't get very many out of the other end. Okay, so when you look at the kind of compare visually the egg and the sperm, remember that those eggs, the viable ones, they really hold on to as much cytoplasm as they can at the expense of the other one. So I like to really think of it as you're putting all of your eggs in one basket. What I mean by that is it's better to have one big, high quality, lots of cytoplasm, gonna last a long time egg than it is to make a whole bunch of it. And if you think about the journey that egg has to make all the way, through the uterine tubes and all the way there, embeds in the endometrium, that whole process, you really want to make sure that they have all the machinery and all the cytoplasm and everything that they need to make a really long trip. Okay, so you really want to emphasize quality over quantity. But for the males, if you think about reproductive strategy for males, it really is just about making as many sperm as possible. And not only that, making them small. And the reason why we want them small is because their job is to travel. You really want those sperm to travel as far and as fast as possible. So you're going to give them very little cytoplasm. And in fact, most of their cytoplasm is going to be shed off of them. Okay, look how tiny their little heads are. And then you want to give them instead a really long, agile tail. Okay, because the fastest sperm wins. So for males, you really want to spit out as many sperm as possible, as many fast sperm as possible. And that's because it's about traveling a distance. Once the sperm reaches the egg, the only thing that has to happen is it has to give off the genetic material, and then it's done. Okay, so the egg has to, it has to undergo all of those divisions to make, as the embryo develops, it has to travel and it will become the fetus. Whereas the male, all it has to do is deposit the genetic material. That's it. So if you think about your cytoplasm from your cells, so if you think about, for example, your mitochondria actually have their own little DNA in them, actually. And your mitochondria came from your mother. Okay, so your mitochondrial DNA came from your mother, not from your father, right? And it has to do with the fact that the cytoplasm comes from the egg, not the sperm. So anyways, I'm kind of going off on a long tangent here, but I mostly want to talk about the two different types of strategies here. One is make very few of them, but make them really, really good. And the other one is make a whole bunch of them, make them really, really fast, but we don't need to make them last very long. Okay, so male sperm are not very short-lived. They don't live very long. They're very short-lived because they don't have the cytoplasm to make the journey. So get to the egg, deposit it, you're done. Whew. Okay, I talked way too much about that, sorry. Okay, so let's talk about some hormonal control of the ovarian cycle. So I think I'm going to stop and restart.